spoke about baroreceptors, right, for example, and basically if there's hypotension, right, the response via those baroreceptors convey signals back to the medulla, which in this case, if it's hypotension, will trigger a sympathetic response. If it's a hypertension, it'll trigger a parasympathetic response. And remember, of course, the whole idea has to do with whether it's an increase in, for example, cardiac output and resistance, or a decrease in cardiac output and resistance. Remember those tables. Another topic was looking at the capillaries. One of the things that occurs at capillaries is, of course, dual mechanisms. We have movement of material out of the capillaries into the tissue and movement of waste from the tissue back into the capillaries, kind of opposing forces. Understanding is what regulates this. Remember, filtration is the movement of material from the capillaries into the tissue. And what governs that is the balance between hydrostatic pressure, which is going to tend to force material out of the capillaries into the tissue, and osmotic pressure, which is going to draw fluid in. The pulling of fluid back into the capillaries is called absorption. The movement of fluid out of the capillaries is called filtration. So I had a couple of diagrams of that. Going back to kind of how the heart works and things like that, we did talk about the importance in maintaining cardiovascular function, or to maintain an efficient cardiac output, you need blood coming back to the heart, which kind of makes sense. If the supply isn't there, you're not going to be able to pump much blood out. So what we have what's called venous return, and remember we spoke about things like the respiratory pump, the skeletal muscle pump, the valves, how they contribute to ensuring blood gets back to the heart by way of venous return. Blood vessels. So, the we have different classes of blood vessels: arteries, veins, venules, arterioles, and capillaries. The layers of tissue, 3 layers versus 2 versus 1, and how that correlates with their function. Remember, with capillaries, the single layer allows for very rapid diffusion, whereas in the case of the arteries, the very thick muscular wall, along with the elastic fibers, allows for the expansion as pressure increases, and basically the recoil to help push blood downstream. So just kind of understand that. So that's going to be it as far as questions related to the cardiovascular system. Any questions regarding that? Yeah.
So just to be clear, blood vessel histology is like the tunica media interna.
Correct.
And we know the functions of those.
Right. Which layers are present in which blood vessels, and how those different layers contribute to the function. Exactly. All right. With respiratory, actually, Pessoy's law is not really respiratory, I just realized. Well, anyway, remember Pessoy's law regarding to blood flow. That really should be on the other one. But blood flow, remember that the factors that blood flow is dictated by, of course, the radius of the blood vessel, the pressure gradient, the viscosity, stuff like that. So basically, you'll need to know what, I'm not going to ask you to calculate anything, but you do need to know the relationship of the different factors and which ones will increase blood flow, which ones will decrease. With respiratory, again, only a few things. The importance of the law of Laplace, and of course, law of Laplace, we started to see that when we were talking about compliance of the lungs. With inhaling and exhaling, the alveoli during inhaling will expand, right? And then when we exhale, the alveoli get smaller. Well, this could potentially present a problem because there is fluid that lines the inside of the alveoli, and as the walls of the alveoli get closer and closer, the attraction of water for other water molecules can cause this sort of a collapse. And as a result, the alveoli would not be able to reinflate. So as fortunately, of course, we have what's known as surfactant that prevents that from happening. And what prevents the alveoli from collapsing, of course, the whole law of Laplace, which I thought I had a marker. There it is. Remember, the law of Laplace basically says that pressure in an alveolus is equal to 2 times the surface tension over the radius. And so, remember I mentioned that the larger the radius of an alveoli, assuming the surface tension is the same, will have a less pressure. So basically what this is saying is that the larger alveolus has less pressure. So normally the small alveoli will collapse into a larger one because there's more pressure in a small alveolus, because that small number results in a big number overall. So air would move from a small alveolus into a large alveolus. The small alveoli would collapse, not be able to reinflate. Fortunately, with surface tension, that equalizes the surface tension, and as a result, we get equal pressure, so there's no movement of air from one alveolus into the other. So that's the law of Laplace. We spoke about respiratory pathologies. Things like, we spoke about emphysema, we spoke about what else? Asthma, we spoke about fibrotic disease or pulmonary fibrosis. So you should know the characteristics of those. So remember, with emphysema, there's really the result, this is a disease that really is primarily caused by inhalation of smoke or the substances that are in cigarette smoke. That damage the alveoli. The alveoli gets damaged and scarred, which makes breathing very difficult. With pulmonary fibrosis, remember that's primarily due to exposure to coal dust, and this causes a scarring of the lungs, which normally the lungs are very elastic, but they have scarred tissue which prevents them from inflating. And of course, we also spoke about asthma as well. So kind of know the characteristics of each of those pathologies. Finally, CO2 transport. We already covered oxygen transport. Remember, CO2 is transported in a different way than oxygen. Like oxygen, it does get transported by hemoglobin, but proportionally not as much. Most of the transport of CO2 is in the form of bicarbonate, which of course acts secondarily as a buffer, which helps to maintain the pH. So kind of know the different ways that CO2 is transported. All right, so that takes us through all the past stuff. Well, it's all past stuff, but I mean really past stuff from the previous exam. Now that really the focus, like I mentioned, the bulk of the exam is going to be on the urinary system. And we started talking about the different components of the urinary system. So, for example, the urinary system is made up of the kidneys, the ureters, one for each kidney, the bladder, and then the urethra. And of course, when we talk about those different structures, 2 of them we spoke about quite a bit, right? The kidneys, because that's really the workhorse of the urinary system. That's where all those different transport mechanisms, the formation of urine, all those things take place. The ureters and the urethra are principally just tubes that transport the urine from one site to the other. The other area, of course, was the bladder, that very muscular organ where urine is stored. We spoke about the micturition reflex, which we'll kind of address in just a second. All right, so like I mentioned, the kidney was the major focus. You should know the anatomy of the kidney in the sense that it's made up, the basic functional unit of the kidney are what are called nephrons. And there are millions of nephrons within each kidney. And remember, those nephrons include, for example, the glomerulus, the capsule, the proximal tubule, the loop of Henle, all those distal tubule collecting duct. There's millions of those throughout the kidney, and that's where all the different processes of urine formation and maintaining a blood volume and things like that occur. Speaking of maintaining, yes, question?
I was wondering, do we have to know like a specific anatomy of the tubules? Cause like I saw in the PowerPoint it says like how each tubule is different, has like a mitochondria and like microvilli. Do we have to know like the specific, or we just need to know like the functions of like the leukopendy and like proximal tubule?
Correct. So know the functions, but you're absolutely right. There, if you were to slice through like different parts, like the proximal and the distal tubule, they look different, but you don't have to go into that detail. It's more function. That's a good question. Speaking of functions, so remember, in addition to the role of forming urine, of course, the formation of urine is important with the kidney, but remember I mentioned it also plays a role in maintaining pH, maintaining blood pressure. Remember we spoke about all those different feedback mechanisms, the myogenic feed reflex and things like that. So know all the functions. Renal blood supply. So remember we spoke about that the kidney is supplied by branches from the aorta known as the renal artery. Once the renal artery reaches the kidney, remember it branches out into different smaller arteries, segmental arteries, right, interlobular. So you should know those arteries and kind of the order in which, not only the arteries and the veins, and there was that nice little chart that I showed you in lecture. So you should know kind of the order of how blood goes through the kidney. All right, so there's three main mechanisms that occur in the kidney. Remember that lead to the formation of urine. I mentioned filtration, reabsorption, and secretion. Secretion I mentioned really in name only, but we really didn't go into details about it. So really the main focus is going to be on filtration and reabsorption. And of course, each of these different processes occur in different places of the nephron. Filtration occurs, remember, right within that what we call the renal corpuscle, or where Bowman's capsule and the glomerulus are found. Remember, blood travels in through that afferent arteriole, gets squeezed across the capillary, the filtrate goes into the capsule, where then it travels into the proximal tubule. Filtration, of course, requires a certain amount of pressure. Much like when we were talking about capillaries under the cardiovascular system in terms of absorption and filtration with the balance of pressures. With here, we also talk about the balance of pressure. So remember, we've got, if you want to filter blood, you've got to have a driving force that's going to push it in this direction. And remember, the driving force is hydrostatic pressure, or the blood pressure, that's going to push it this way. But we've also got some opposing pressures. We've got the osmotic pressure, which is the proteins in the blood that will tend to draw fluid back. And then we've also got the buildup of fluid within the Bowman's capsule. As blood is being filtered, right, there's fluid here, and that fluid can actually kind of push back. So what we want overall is that the hydrostatic pressure needs to exceed the sum of both the capillary osmotic pressure and the capsular pressure. And I had a couple of diagrams about that. All this filtration occurs across a membrane, much like when we were talking about, remember with the alveoli, the respiratory membrane, you've got the simple squamous cell, in the case of the respiratory, from the alveoli, the simple squamous cell of the capillary, and then the basement membrane in between. Same thing is really true here, where we've got the endothelium, or the simple squamous cells of the glomerulus, right? And then you've got, but in this case, you've also got what are known as podocytes. These are these cells that wrap around the glomerulus, and they create little pores or slits, and so things are kind of go through a sieve. So we've got the capillary endothelium, the podocytes, and we got the base and membrane in between. So very similar type of arrangement. I mentioned certain definitions or certain ways that you can evaluate kidney function. We spoke about, for example, what's known as filtration fraction. So you should go back and see what the terms, what those mean. We spoke about GFR, or glomerular filtration rate. And remember, this is the rate that filtration occurs. And of course, it's a way of assessing how well the kidney is doing. Much like with the heart, we assess how the heart's doing by, what's the cardiac output. In the kidney, we're looking at how much filtrate is formed over a unit time. And remember, GFR normally is about 180 liters per day, is squeezed across the glomerulus, goes into the tubules. But of course, as we know, roughly about 99% of that is reabsorbed. Some other terms that we mentioned, renal threshold. So renal threshold, so remember I mentioned that we've got, all the solutes in the blood, glucose, amino acids, ions, of course water, filtered across the glomerulus, go into Bowman's capsule, go into the tubules, right? And normally, all of those are reabsorbed. You normally, if you were to do a urine sample, you would not see any glucose or amino acids or anything. They all get reabsorbed. However, there does reach a point where there's too much of those solutes, and then they end up staying in the urine. That's the renal threshold. I use the example of when we're talking about reabsorption of glucose. Remember I mentioned that, glucose, if your blood glucose level is a certain amount, that or below that amount in the blood, you're going to reabsorb all of it. If you exceed that amount, like if you're diabetic, if you're hyperglycemic, you can't reabsorb all of it. It's impossible to reabsorb all of it, so what's left spills over in the urine. I use the example of a dam where water is held back, but if that water reaches a certain level, it spills over. So that's kind of what we're talking about. We spoke about renal blood flow. Renal blood flow, of course, is related with those afferent arterioles. The afferent arterioles increase, when the diameter increases, that allows for a greater blood flow. If they're constricted, that decreases blood flow. Filtration pressure, again, that's all kind of related with the factors that regulate GFR, right? So you've got three factors that go into making up filtration pressure. You've got the hydrostatic pressure, which is forward moving. Then you've got the osmotic and the capsular pressure, which are opposing. So you want to make sure you've got the forward pressure exceeds the two opposing ones. All right. What else? Okay, so not in any particular order. I guess what I'll do is I'll talk about regulation of GFR. So one of the things I spoke about is how important, for the most part, GFR is maintained at 180 liters per day. But this isn't done magically, right? We have mechanisms that can respond to changes occurring elsewhere to ensure that the GFR doesn't get too high, which can damage the kidney, or too low, which can also lead to kidney failure. And one of the ways this is done by that myogenic response. So remember, the myogenic response is where you've got, say, if the blood pressure goes up, normally if you increase, high blood pressure is going to increase the blood flowing through the, going through the glomerulus. And if it gets too high, this could damage the capillaries and everything. So what normally happens, remember with the myogenic response, is the afferent arterioles will expand, and then they constrict and get smaller, which reduces the blood flow to stabilize so the pressure is not as high going into the kidneys. It reduces the blood flow. That's that myogenic response, to ensure that when blood pressure goes up, this is an adaptation to ensure that high blood pressure is not going to be translated into too high blood pressure going into the kidneys where to damage it. The other factor, the other strategy for regulating GFR is a little bit further downstream, and that's where we talk about the tubuloglomerular feedback, and that all kind of relates to this. So remember with the tubuloglomerular feedback, so you've got fluid that's filtered across the filtration membrane, goes into the proximal tubule, through the loop of Henle, and then you've got, then reaches the distal tubule. Well, the distal tubule has these cells called macula densa cells, which are chemoreceptors. Much like chemoreceptors work in the respiratory system with sensing CO2, pH, things like that, changes, these chemoreceptors will sense ion concentration. And if the chemoreceptors in the distal tubule, known as the macula densa, pick up that the sodium levels are too high, that's an indication that the GFR is too high. So what happens is those macula densa cells, remember, secrete what was shown on the slide as a paracrine, but now we know that it's actually adenosine. Adenosine is secreted by the macula densa, targets these granular cells which surround the afferent arteriole and causes them to constrict. So whether you're looking at the myogenic response or the tubuloglomerular feedback, both of these responses are going to are responses to high GFR to help to bring it down. One is more direct on the blood vessel. The other is kind of indirect. First, you're sensing that there's too many ions, too much GFR, and then the signal is sent out on those granular cells to cause constriction of the afferent arteriole. The juxtaglomerular apparatus is just another name for the macula densa and the granular cells combined, that whole area. All right, so we just mentioned that what we just spoke about was the regulation of GFR. So that takes us pretty much through the filtration part of kidney function. Then, of course, we spoke about reabsorption, and we didn't cover every single ion, every single molecule that's present. I focused on some of the key ones, and some of those are things like sodium and glucose. So you should know how sodium gets reabsorbed. Remember, I mentioned that it can be reabsorbed directly, just on its own, across the apical membrane. Remember, there's two membranes of a cell, or, well, there's four sides, but there's the apical and basal side. Going from the kidney lumen, it gets pulled in via the apical side, goes through the cell, and then is pumped out into the blood. You should know the different pathways. We spoke about active transport, facilitated diffusion. I also mentioned that sodium can also get pulled across kind of in a co-transport way with glucose, what we call the SGLT. And of course, glucose moves out through facilitated diffusion, things like that. So those are really the main molecules that we spoke about. But of course, one thing we can't forget is water, right? We spoke about ions, glucose, movement of them, but of course, water's always moving too. And how much water we excrete and how much water we retain is dependent upon the endocrine control, or the role of endocrine organs or hormones. And of course, how much water we retain or how much water we excrete can in turn affect blood volume, right? The more water we retain, that's going to increase blood volume. The more water we excrete, that's going to reduce it. And the hormones that we mentioned, of course, were ADH. You remember the other name for ADH? Vasopressin, exactly. So in case on the exam I write vasopressin, I just want you to remember that ADH and vasopressin are the same. Those other hormones that you guys mentioned, of course, are two different ones, right? We got A and F, and we've got aldosterone. So what does ADH do? What happens when ADH is secreted? Where does water go? Do we dump water out or do we retain it? We retain it. So this is produced in response to dehydration. You guys haven't done the urine lab, have you yet? Okay, so yeah, that's coming up. Everyone in this whole building knows when the urine lab is because you'll see people walking around with these little Dixie cups, walking around and try not to bump into them. So when you're dehydrated, you produce ADH, which, remember I mentioned, activates aquaporins. Remember, these are these water channels that pull water in. So you should know what's produced under what conditions. So when the water levels are low or low blood pressure, we want to retain water. That's when ADH is produced. When sodium levels are low, we produce aldosterone. And aldosterone, remember, instead of stimulating production of aquaporins, we see the activation of the sodium potassium ATPase. So that's the mechanism, right? ADH pulls in water. Why? Because we're making aquaporins as a result, which pulls water in. Aldosterone results in the retention of sodium. Why? Because aldosterone is stimulating the production of the sodium potassium ATPase pump, which pulls sodium in. So those are factors, conditions that result in elevated sodium, elevated water. But say, for example, what if we have too much water, or more than enough water, more than enough sodium? That's where we see the role of ANF. And where is ANF produced? Where is that hormone made? The heart, exactly. And so with high blood pressure, high sodium, high fluid levels, ANF is produced, which blocks ADH, which blocks aldosterone. So if you no longer have the hormones that are allowing for sodium retention or water retention, by default, more is excreted. So it actually has a diuretic effect. So that's what ANF is. I briefly mentioned pH. One of the functions of the kidney is pH, and there's a couple cells in the collecting duct. You saw in that diagram the A cells and the B cells. And one of the things, of course, is if your blood pH or if your body pH is too high, let me rephrase that. If your pH is too low, it means that you're in an acidotic condition, right? A lot of hydrogen ions floating around makes it acidic. Then you've got cells of the kidney that will pump hydrogen ions into the tubule where it'll be excreted. So essentially, if there's too much acidity, too much hydrogen ions, the kidney gets rid of those hydrogen ions. If you're in an alkalotic state, pH is too high, we pump out bicarbonate. And obviously there's a bunch of mechanisms on those charts. We didn't really get into those. I just kind of want you to know kind of the end game of what happens. Finally, the micturition do you have a question?
So you do not need to know about the carbonic anhydrase?
No. I mean, that's an important enzyme, but we really didn't go into that. Yeah. Micturition reflex. So this is, again, the micturition is the process of urination. And that, of course, depends on the sensation that we have to go. So if you remember with micturition, what happens is, so what are the structures involved? We've got the bladder, right? And then we've got those sphincters, the internal and external urethral sphincters. So those are the players. We also have some nerves. We've got autonomic nerves, of course, and then voluntary nerves. So how does this all work? So what triggers the micturition reflex is, of course, your bladder is filling up with urine. And remember, the bladder has got this thick muscle. When the bladder is filled with urine, the bladder stretches. And we have stretch receptors in the bladder wall that detect that stretch. Those stretch receptors fire signals to nerves that are going up the spinal cord and to the pons. So that's the first step. Stretch activation, sending a nerve signal up to the pons and the spinal cord. Those areas are what we call the micturition center. And what happens is then we've got, this activates the parasympathetic pathway, which sends signals down, and the parasympathetic neurons innervate two main areas. They innervate the bladder wall, where the muscle is, and they innervate the internal urethral sphincter. So remember, parasympathetic is part of the autonomic nervous system, which is involuntary. So this is not anything we have control over. This is going to happen whether we think about it or not. And what's going to happen is those autonomic neurons are going to come back to the bladder, and they're going to cause that muscle to contract, which is going to squeeze the urine in that bladder. And then at the same time, the autonomic neurons are also stimulating the opening of the internal sphincter. So in response to the stretch, you get a contraction of the bladder and an opening of that sphincter. Okay, so that's the involuntary part, and that's when you have the sensation you've got to go, right? Yes?
The detrustor muscle is the muscle that squeezes the
Correct,
bladder?
yeah, so the muscle in the bladder wall is called the detrustor muscle, exactly. All right. So now from there, what happens is then, so that you have the sensation to go. Well, one thing that I've left out, of course, is the external urethral sphincter, right? And that's the last part. Normally, as post potty trained people, right? We have the ability to, for the most part, kind of, even if you have to go, we're able to kind of control it until we find a bathroom. Obviously, there are exceptions, like if you're ever stuck on a freeway for an hour and I won't go there. Anyway, but anyway, I guess I just did. Anyway, so what happens is there's a somatic neuron that innervates the external urethral sphincter, and then normally it's closed, but what happens is with our voluntary process, whatever, the nerves will stimulate it to open up. And that's where we go to the bathroom. With infants that aren't potty trained, what happens is we don't have that ability to keep it closed, right? It's always open. As any of you with kids or nieces or nephews or whatever, you know that basically whatever goes in just comes right out right away, right? So, all right, so those are the topics. As far as the short answer, yes?
So, I mean, it's about like the juxtaglomerular apparatus. So I know how granular cells secrete renin. So do we need to know about like the renin and angiotensin system?
Right, so the formation of the angiotensin, yes. So what happens, yes, so you've got the renin, which stimulates angiotensinogen to angiotensin 1, and then you've got ACE, yes, which ultimately leads to the formation of aldosterone.
Yeah, another question is for the Victorian reflexes, do you have to know about the voiding reflex and the guarding reflex as well?
No,
Okay.
not at all. You're a deep thinker, I can tell. Yes?
So according to the pH, I mean the kidney function, do we have to know like the stages, like alkaloid stage, like whether when the pH level is low and high, which stage it goes through, or we have to just know the functions like osmolarity and pH and all that?
Yeah, so what you need to know is that, so when you're in a alkalotic state, which means that the pH is too high, the response of the kidney. So when the pH is too high, those cells of the collecting duct excrete bicarbonate. When the pH is too low, high acidity, you've got too much hydrogen, you excrete the hydrogen. So that's the extent.
Is it okay, like, are we okay, to know the hormones and like what they do, or do we need to know like each step, along the way?
I'm not sure what
Like,
you mean.
For example, like, vasopressin, binding, aldosterone, the steps of aldosterone when it's secreted. Do we need to know every step, or do we just need to know like what response they have, like what they're for?
Okay,
what they do?
so what you should know, so there's that chart where it talks about, for example, how the hormones work, like what, for example, whether they, I think there was the one where, how does ADH work, and ADH works,
Like transcription of nuclease,
I'm sorry. Yeah. So when you're looking at, so vasopressin binds to vasopressin receptors.
yeah, like.
Yeah, and then that activates expression of the aquaporins.
Yeah.
Yeah, that's what you need to know. I mean, if
So
there's
we need to know
anything
every
in,
step?
what?
We need to know like all the steps. You're telling me like we need to know all the steps for like.
Well, whatever we
for the,
discussed, I mean, for example, I mean, like what I just mentioned was that, like, if you look at the chart you're looking at, which other ones, like, come to mind? I mean, so binds to a receptor. I mean, certainly there are things like, yes, transcription, translation, but in that respect, what I'm looking at is really the end game. Vasopressin binds to its receptor, and the result is increased expression of aquaporins.
Oh, okay. So we're good with just that.
Yeah.
Okay.
Now, as far as other things, I mean, I don't want to get into trouble if I say you don't need to know it, whatever. But basically, you know, whatever we covered in class, you should know. I know that really helps a lot, right? Short answer topics. Okay, so I made it very general for the sole purpose that, you know, because I hadn't exactly made the questions yet, but I just wanted to get you thinking. So, question about reabsorption. Again, you should know the mechanisms of how things are reabsorbed. For example, you know, we spoke about things like facilitated. How does glucose get absorbed? How does sodium get absorbed? You know, facilitated diffusion. Is it simple diffusion? Is it active transport? You know, how is it? Which membrane? Remember, so if you think of membranes, the membrane that's closest to the kidney lumen is called the apical membrane, and so fluid or solutes would get pulled across that into the cell, and then from the cell, those same solutes are transported across the basal membrane into the blood. So, just those terms. Filtration. I mean, that's kind of a really open-ended question. You know, obviously, where does filtration occur? I can ask you a question on what the filtration membrane is. You know, where does it occur? How much gets pumped out into the blood? What is filtration pressure? What is GFR? Endocrine regulation of blood pressure. All right, so now when I say blood pressure, really what I'm referring to is this part right here. So it's really those hormones. You know, because really, even though we're looking at their effects on the kidney, it's ultimately causing an effect on the blood. Because if you move water from the kidney back into the blood, that's going to increase blood pressure. If you move sodium back into the blood, that's going to increase blood pressure. If you move sodium and water out, that's going to decrease blood pressure. Micturition reflex, there'll be a short answer question on that. Question on the blood flow through the kidney. You know, I might ask you, you know, just to describe kind of like a flow chart of blood flow from aorta to the renal artery. And that would be both arteries and veins. So getting back to a question that was asked in the beginning about 40 or like 48 questions, after class I've got to go through and take a look and see. I'll let you know like ASAP. I'm not sure exactly, but that's